Edvotek Informational Video, Testing for COVID-19. Coronavirus 2019, or COVID-19, is a respiratory disease caused by the novel virus SARS-CoV-2. This infectious agent emerged in December 2019 and spread worldwide in a very short period of time. Coronaviruses have a single-stranded RNA genome wrapped in a helical capsid. A membrane envelope surrounds the capsid. The envelope is studded with spike proteins that help the virus infect cells. By electron microscopy, the spike proteins form a diffuse ring around the virus particle. This trait led to scientists naming these viruses using the word corona, Latin for crown or halo. Respiratory viruses, like SARS-CoV-2, spread person to person through liquid droplets formed when you cough, sneeze, talk, or breathe. The largest particles, droplets, don't last long because they are too heavy to remain in the air. However, smaller droplets, suspended in the air as aerosols, can travel a further distance to infect people. Soap, hand sanitizer, and other disinfectants kill coronaviruses, so frequent washing of hands can limit its spread. Touching your face with contaminated hands can introduce the virus to your mucous membranes, so it is important to keep your hands away from your eyes, nose, and mouth. A high-quality mask which covers the mouth and nose prevents respiratory droplets from spreading via cough or sneeze. Symptoms of COVID-19 include fever, cough, and shortness of breath. In severe cases, patients develop pneumonia, respiratory distress, or kidney failure. Conservative estimates show that 25% of COVID patients exhibit symptoms for over 30 days post-infection. Sadly, this infection can be fatal, even in healthy people. If you are exhibiting symptoms, contact your healthcare professional for guidance. There are two diagnostic tests to confirm an active COVID-19 infection: the Nucleic acid amplification test and COVID Antigen test. A third test, the COVID Antibody Test, determines whether a person had been previously infected by the virus. A positive test does not mean that a patient will become seriously ill. However, these diagnoses are important as they allow epidemiologists to trace the spread of COVID-19. The Nucleic acid amplification test, or NAT, is a molecular test that detects the RNA genome of SARS-CoV-2, which signifies an active infection. Because this test is extremely sensitive and can detect minute amounts of the virus, it is an ideal assay to diagnose SARS-CoV-2 infections. NAT uses the polymerase chain reaction, or PCR, to detect the viral genome in a sequence-specific manner. However, PCR only works with DNA as the starting material. To address this, total RNA is extracted from the patient sample and converted to complementary DNA, or cDNA, using reverse transcriptase. The cDNA can then be amplified using PCR. The amount of viral cDNA in the sample is measured using quantitative PCR, or qPCR. This technique measures the amount of cDNA in a sample at the same time it is amplified. This is accomplished by adding a dye that only fluoresces when bound to double-stranded DNA. The fluorescence of the sample is measured after each round of PCR. An increase in fluorescence is directly correlated to the increase in double-stranded DNA in the sample. A positive result is determined by the CT value, which is the threshold for amplification. Low CT numbers suggest a high amount of starting viral RNA because the sample hits the threshold faster. The NAT is considered the gold standard because it can detect very low amounts of virus in patient samples, even in patients without symptoms. However, the tests are expensive and can take a long time to get results. The COVID Rapid Antigen Test is a highly sensitive enzyme-linked immunosorbent assay. This test uses antibodies to detect the presence of SARS-CoV-2 surface proteins in patient samples. Antibodies are Y-shaped molecules that specifically identify antigens within complex mixtures. COVID rapid tests use antibodies that react with specific viral protein fragments found in patient samples. Antigen-antibody complexes are assembled in samples where the viral protein fragments are found, resulting in a positive result when a detection substrate is added. For the rapid test, the antibodies are embedded in a test strip. The patient sample is added to the well, and it travels up through the test strip. If any viral proteins are present, a colored line will form at the T, or test line. To make sure the test is working properly, each test has a control line which changes color regardless of the outcome. These rapid tests are easy to perform and can provide results in 15 to 30 minutes. However, they are not as accurate as the NAT, especially early on in the infection when virus levels are low. Once the patient's immune system has cleared the SARS-CoV-2 infection, no viral RNA or protein remains in the body. However, the antibodies generated to fight off the infection remain in the body after the patient has healed. The COVID Antibody Test detects the presence of antibodies to SARS-CoV-2 in patients, signifying a previous infection. Most antibody tests identify IgM and IgG in patient samples. The IgM antibody serves as the first line of defense against the SARS-CoV-2 virus, and the IgG antibodies are produced in the plasma B cells as part of the adaptive immune response. A special technique called a sandwich ELISA is used to identify these antibodies. SARS-CoV-2 antibodies and antigens are bound to a reaction vessel. The antigen captures any IgM or IgG present in the patient sample, which will cause a positive result when the substrate is added. By using this assay, researchers will be able to identify individuals affected by this disease who were not tested while ill. However, since the body takes several days to produce these antibodies, the ELISA cannot diagnose infection before clinical symptoms arise. Please remember, this video does not substitute for guidance from a healthcare professional. If you are exhibiting symptoms of a serious illness, please reach out to your doctor or your local public health officials for advice.